Streps and enterococci are amongst the most common pathogens encountered in both human and veterinary medicine. Streps are notorious for the post-infection sequelae which sometimes occur and can be life-threatening. In today's lecture, we'll be talking about a group of organisms which are recognized to cause a wide variety of infections across veterinary species and into people as well. These organisms are all classified as biocontainment level 2, and like our staph, they are gram-positive cocci. They're also aerobic or facultatively anaerobic, and we commonly group them based on either their hemolysis patterns, so alpha streps or beta streps, and their Lancefield groups, which is based on surface antigens that can be identified serologically. Streps can be fastidious, requiring either blood or serum in the media to grow, and so they don't always grow on regular blood agar. We can easily differentiate streps and enterococci from the genus Staphylococcus using the catalase test. Here we can see a typical gram stain of Streptococcus dysgalactiae, and while these are gram-positive cocci just like staph, I'd like you to pay attention to their arrangement. Here in this image of Strep equi subspecies zooepidemicus, you can see a long chain of organisms forming a line, as compared to Staphylococcus, which forms these grape-like clusters. So remember, streps form chains, staph form clusters. Here we have a culture of Streptococcus agalactiae on blood agar. On the right-hand side, I think you can appreciate a small zone of beta hemolysis surrounding the colonies. Here we have a strep canis with a much brighter zone of beta hemolysis. And finally, a strep equi subspecies zooepidemicus with very pronounced and obvious beta hemolysis. Strep dysgalactiae is characterized as alpha hemolytic, and I think you can appreciate the discoloration of the blood agar under and surrounding the colonies. Remember, alpha hemolysis or partial hemolysis is actually the presence of methemoglobin within the unlysed erythrocytes. Similarly, strep suis is also alpha hemolytic. And then finally, Enterococcus species, in this case Enterococcus faecalis, is non-hemolytic and produces these characteristic blue-gray colonies. Our streps and enterococci are all host-associated, and they're all part of the normal microbiota. Our streps tend to be found on the mucous membranes and respiratory tract, while the enterococci, as the name would suggest, are found in the intestinal tract. Carriers of these organisms are really the primary source of infection for the broader community, although environmental contamination with Enterococcus species is associated with nosocomial infections, although this is more commonly recognized in human infectious disease than in a veterinary context. There are 116 species of Streptococcus and 62 species of Enterococcus. Like I mentioned earlier, we can differentiate both of these genera from Staphylococcus using the catalase test, where our staph is catalase positive and our streps and enterococci are catalase negative. We can then differentiate streps from enterococcus using either the Escalin test or their ability to grow in high concentrations of sodium chloride. Enterococci is halophilic and will easily grow. Our streps can then be further differentiated by hemolysis type, as I mentioned, or Lancefield grouping followed by other biochemical tests. You may have heard of group A strep in the media. This is the Lancefield group of strep pyogenes, a very common and important human pathogen. Biochemical identification of streptococci is certainly possible. On the left here, you can see an image of the API 20 strep kit, which was a commercially prepared set of biochemical tests, sort of miniaturized. So in each well, we had a different reagent. We would make a suspension of our bacteria, aliquot that into these wells and incubate it, and then look for a colorimetric change, which was recorded on this sheet, and then the results looked up in a database. In the CAMP test, we're looking for an interaction between our test organism and Staphylococcus aureus and the enhancement of the double zone of hemolysis surrounding the staph. So in this image here, you can see we have a streak of Streptococcus agalactiae with greatly enhanced hemolysis surrounding the staph here. And then we have a strep uberis on the right here, labeled B, where we have no enhancement. Strep agalactiae is CAMP positive, strep uberis is CAMP negative. Lansfield grouping is named after Rebecca Lansfield, who first developed this assay. We have a number of different groups. A, B, C, F, and G are polysaccharides on the cell surface, while group D is lipoteichoic acid. Some organisms are known to not group reliably with the Lansfield antisera, notably Strep suis, which is a very important pathogen for pigs and also a critical zoonosis. Just like the staphylococci, streps produce M scrams. They're also famous for superantigens. So strep pyogenes produces an exotoxin superantigen that's responsible for necrotizing fasciitis and toxic shock syndrome in people. Some Streptococcus canis produce a phage-mediated superantigen, which I'll go into more detail later on. They're oftentimes encapsulated, which prevents phagocytosis. Notably, some capsules which are composed of hyaluronic acid are indistinguishable from certain host tissues, and the immune system's response to these capsules may explain the post-infection autoimmunity and those potentially lethal sequelae. Strep produce hemolysins, which are cytotoxins active against eukaryotic cells, both erythrocytes and other cells, and then camp factor, which is a pore-forming toxin that we saw demonstrated a couple of slides ago. Strep pyogenes is a pathogen that commonly infects people, and some have even hypothesized that it may have impacted some important figures in history, notably the composer Wolfgang Amadeus Mozart. Now, the death of Mozart is something that was really the subject of a film from the early 1980s called Amadeus. This movie reimagined a scenario surrounding Mozart's death, focusing on the relationship between Mozart and his contemporary Salieri, who was very jealous of his talents. Now, in the movie, Salieri schemed to drive Mozart mad by pretending to be essentially his dead father, and Mozart dies of exhaustion while writing his Requiem death mass. But what really happened? Well, researchers have looked into this, and by evaluating death records from the time, they have a much better idea of what actually happened. So Mozart is known to have died on December 5th, 1791, and the movie was right in that he was working on his Requiem death mass on his deathbed. Records from the time describe a localized epidemic of deaths involving edema. And earlier that fall, Mozart had reportedly had an inflammatory fever. These researchers speculated that Mozart may have suffered from strep throat, which is caused by Streptococcus pyogenes, followed by post-streptococcal glomerulonephritis, so an inflammation of the kidneys, which is an autoimmune response, perhaps secondary to the production of cross-reactive antibodies that the body was making to target the strep pyogenes. The Requiem Mass is an epic piece of classical music, and I'll put a link above to a reimagined version in sort of a rock opera format. So strep pyogenes, as we've talked about, causes strep throat. It's been associated with necrotizing fasciitis and scarlet fever in people. Streptococcus pneumoniae causes opportunistic infections and skin infections in people. In horses and guinea pigs, we also see pneumonia. Strep agalactiae causes chronic mastitis in cattle, while dysgalactia, subspecies dysgalactia, causes acute mastitis. Streptococcus equi, subspecies equi, is the agent of strangles in horses. And strep canis causes a variety of infections in dogs and cats, from septicemia, wound infections, abscesses, and respiratory tract infections. Strep equi, subspecies zooepidemicus, can also cause infections in horses, although they're typically more joint infections. In cattle, it causes mastitis. In pigs, there seems to be an emerging syndrome of septicemia. In poultry, we also see septicemia, and in dogs and cats, we can see respiratory tract infections. Streptococcus suis is a very important pathogen of pigs, causing meningitis, arthritis, and septicemia, and a very important potential zoonoses associated with similar diseases in people. One important sequela of strep suis infections in people is actually hearing loss. Strep bovis in cattle is responsible for rumen acidosis, and it causes opportunistic infections in other species. Finally, Enterococcus species causes opportunistic infections in a variety of animals. Starting off with strep agalactiae and mastitis in cattle, This is a cause of contagious mastitis, meaning that infected cows are the source for the herd as a whole. This is mainly subclinical disease with bouts of illness, and the animals, generally speaking, are not systemically ill. Presence of this disease suggests that there's been a breakdown of biosecurity. So perhaps teat hygiene, milk equipment not being cleaned adequately, or inadequate hand washing. Treatment of strep agalactiae mastitis often involves intramammary antibiotics. And unlike staph, many streptococci fortunately remain susceptible to penicillin. Streptococcus dysgalactiae causes environmental mastitis, which isn't associated with a colonized animal in the herd, but rather contamination of the teats from the environment or bedding. This too is oftentimes subclinical and suggests that there's management issues, including the need for improved cleanliness, better bedding, and access to shelter. Strep dysgalactiae mastitis is frequently treated in the dry cow period, including antibiotics and potentially teat sealer. In this cytological preparation of bovine milk, you can see Streptococcus uberis, these nice chains of coccoid bacteria. Streptococcus equi, subspecies equi, cause of strangles, is actually a normal inhabitant of the upper respiratory tract. Strangles, the colloquial name for abscessation of the submandibular and retropharyngeal lymph nodes, is a really serious disease in horses. It's transmitted between individuals through contact with nasal secretions or discharge from abscesses, so essentially ruptured lymph nodes and all of the pus that makes its way into the environment. And this contact can be either direct or through fomites. It's very contagious. There's a high morbidity rate, so up to 100% of animals who are exposed become infected, and a low but not insignificant mortality rate of around 10%. Control of this disease really relies on isolation. You want to prevent transmission between animals, prevent exposure to purulent material, such as you see here. Disease develops following a 6 to 10 day incubation period. Penicillin is the treatment of choice for strangles, and much like strep pyogenes in people, long-term sequelae following infection are possible. These include metastatic abscesses, also known as bastard strangles, which is due to the hematogenous spread of the organism to various body sites, from the lungs, the abdominal viscera, and even the brain. Purpura hemorrhagica, which is a vasculitis secondary to the deposition of immune complexes, which, of course, you'll remember from immunology is known as a type 3 hypersensitivity reaction, and guttural pouch empyema. 75% of horses who recover have enduring immunity to future infections, and there seems to be a transfer of immunity through the colostrum, and so foals generally are protected until weaning. Here we have some pathological images of horses who have had post-infection sequelae. So on the left, we can see gingival petechiation, these little red spots all over the gums, and mesenteric hemorrhaging in a case of purpura hemorrhagica.